DNA is a molecule made up of two strands twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases, which are adenine, cytosine, guanine, and thymine. The two strands are complementary. This means that if there is thymine on one strand, there is adenine on the opposite strand. And when there is cytosine, there would be guanine on the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how two strands of DNA are replicated. DNA replication starts with the unzipping of a DNA strand, and this unzipping is done by an enzyme called helicase. Now we have here a zipper, and we will use this as a model of a DNA strand. Now, assume that this thing is the helicase, and now this would unzip the strand of a DNA. And this unzipping would produce what we call a replication fork. The separated strands each provide a template for creating new strands of DNA. Now here comes an enzyme called the primase, which makes a small piece of RNA called the primer. This primer marks the starting point for the creation of the new strand of DNA. Now here comes an enzyme called DNA polymerase, which will bind to the primer and start the creation of new strands of DNA. Now, what DNA polymerase does is that it adds new DNA bases to the separated strand and will move through the 5' end to the 3' end, thus creating a new strand of DNA. Okay, so the same thing would happen to this other strand here. A primase would first make a primer, and then the DNA polymerase would bind to this primer and start adding new DNA bases. But remember that the DNA polymerase only travel through one direction, from 5' end to 3' end. And this strand here is 3' to 5'. So what happens is that after DNA polymerase adds new DNA bases to this segment, it will travel back here. First, the DNA primase would make another primer, and the DNA polymerase would bind to it again, and then create another, and then add another DNA bases to this segment. So what happens is that it adds new DNA bases in this in this in this in this portion, and then goes back again from 5' to 3' end, and then again in here, and then again in here. So what it creates is what we call Okazaki fragments, since it does not continue creating new strand of a DNA same as in this strand. So this is called the leading strand, and this one is called the lagging strand. Okay, now since this strand is not the same with this leading strand, since the lagging strand is made up of Okazaki fragments, since there are primers in it instead of DNA bases, an enzyme called exonuclease would remove these primers, and then the DNA polymerase would would add new DNA bases to these empty spaces for this DNA strand, thus creating a continuous strand of DNA. Okay, so what happens now is that we are now able to create two DNA strands. Okay, now finally, an enzyme called DNA ligase would seal up the DNA fragments, so we could now have two sets of continuous DNA. DNA replication is described as semi-conservative since each DNA molecule is made up of one conserved DNA strand and a new one.